Tim, for the invitation. So as George said, all of my training has basically been in molecular biology and genetics. So I am a card-carrying molecular biologist, but I've always been interested in evolution. So ever since I was an undergrad, that's really what I wanted to study, is to take molecular approach to understanding the process of evolution. So today I'm going to tell you several stories that we've worked on or are currently working on, and it'll sort of weave a thread through this talk where we'll keep increasing the resolution in which we can observe the dynamics of adaptation using experimental evolution in yeast. So to start really simply, if you have a population and you expose that population to a selective pressure, it will adapt. And populations that are adapting in parallel, so without any gene flow between them, often tend to find the same solutions to, or similar solutions to the same evolutionary problem. So a classic example of this that I like to point to at the beginning of the talk are these cavefish populations. And so you can find these in parts of Mexico, the US Southwest, and there's sort of two different morphologies. You have this surface form that looks like your normal swimming freshwater fish. Periodically, this will find itself trapped in a cave environment, and when it does, it will adapt. So here are two populations on the right that have adapted independently to the same selective pressure. And they've done so through similar morphological changes. So they've lost pigmentation and lost eyesight. And in fact, this parallelism even extends to the genetic level. So both of these populations have different mutations in the same genes underlying loss of pigmentation. And so you have parallelism here at both the phenotypic level and at the genetic level. And so one of the questions that really drives my work and really the whole field of experimental evolution is, how surprising is this type of result? In other words, how predictable or reproducible do we expect evolution to be? And this is a difficult thing to address in natural populations, but it's something that we can begin to address using laboratory experimental evolution. And so this is what we do. There are multiple varieties of how you can do evolution experiments. So I'll sort of walk you through a type of evolution experiment that we might do. And so we use yeast, and we grow our yeast in 96-well plates, so we can have about 100 populations in a really small footprint. And you can imagine doing the following experiment. You can come in and you can inoculate a plate with 96 different initially identical populations. You can let that plate grow to saturation, come back in the next day, dilute all these down into fresh medium, let them grow to saturation again, dilute them again, and just simply repeat this cycle over and over again. And that's essentially what we do in my lab. We don't actually do all of our dilutions by hand, so we rely on robotics. So we have a robot in the lab, and my grad students can come in, or I could come in, and we can set the deck of the robot up with our saturated overnight cultures. We can just hit start, and the robot will dispense all the fresh media. We'll resuspend all of our populations and do 1 to 1,000 serial transfers. And so the combination of miniaturization down to 96-well plates and automation allows us to very easily maintain hundreds of populations and propagate them for thousands of generations. And so it only takes about a half hour or so of actual bench time to transfer 400 cultures in a given day. As we do one of these evolution experiments, we have a lot of tools at our disposal. One of the important ones is that we can freeze all of these populations. So every so often, we'll take all of our overnight cultures, we'll add glycerol, pipette up and down, and we'll just store it in the -80. And so this is our frozen fossil record of all of our evolution experiment. So we can go back to any population at any time point in the past, resurrect it, we can sequence it, we can phenotype it, we can measure its fitness, or sort of restart the evolution experiment from a different earlier time point if we so choose. And I'll give one example of that later. We use flow cytometry extensively in our work, and we use flow cytometry as a way of getting a quantitative measure of fitness. How much better is an evolved strain from its ancestor? So we can take an evolved population, we can mix it 50-50 with the fluorescently labeled version of its ancestor, and then we can just compete those two head-to-head in co-culture and ask how quickly does our evolved strain outcompete our ancestor. And this gives us a very reproducible and quantitative measure of fitness. One of the most powerful tools in our arsenal is next-generation sequencing. And so now sequencing costs keep plummeting. The last time we did the calculation, it cost about $1 to do 1x coverage sequencing of the yeast genome. The catch is you don't do 1x coverage sequencing. You have to do like 4,000x coverage. So you learn to sort of multiplex your sequencing. But it's now quite easy to sequence any of our evolved populations and see exactly how they're changing. Mutation in this particular gene, it's a missense mutation, it's likely to impact function in this way. As George mentioned in the introduction, and I may have mentioned offhand, we do this in yeast. There are a number of reasons why I think yeast is sort of an ideal system for doing experimental evolution. Yeast is a rapidly fast-growing microorganism, so it divides every 90 minutes. We can easily get 10 generations of growth per day. So within a year, you can easily get 3,000 generations of experimental evolution. Yeast can be stably propagated as haploids, diploids, or even tetraploids. And so we can ask directly what's the effect of ploidy on adaptation, and I'll talk a little bit about that later. Yeast can be propagated either sexually or asexual, and so we can use this to start to directly ask questions about what is the effect of recombination during adaptation. We can also use the yeast sexual cycle to do what yeast was really brought in as a model system to do, and that's genetics. And so we can ask very specific genetics questions about our evolved lines. And yeast have a small genome, so they're really amenable to next generation sequencing. And the last reason why I think yeast is a great model system for experimental evolution is that yeast has been a model system for over 80 years now. So we know more about yeast than we do probably any other eukaryotic organism. And so if we find a mutation in a particular gene, chances are there's somebody in the world who's working on that gene. And we have a lot of sort of a rich history that we can go to in understanding and interpreting the results we see. So this is our experimental evolution setup. So what kind of things can we look at when we do an evolution experiment? The first evolution experiment we tried is we just wanted to track a particular phenotype through a population. And so previous work that we had done, we had shown that for a haploid yeast strain that's propagated asexually, there's a fitness advantage to be gained by shutting off the mating pathway. So in other words, there's a cost to producing mating pheromone and to be sensing a mating partner. And so normal yeast will exist in two types, unfortunately named A and alpha. When an A cell comes in proximity to an alpha cell, it will sense mating pheromone produced by that alpha cell. It will arrest its cell cycle and it will form what we call a shmoo, so a mating projection, and these two cells will then fuse at this shmoo tip. This sensing and signaling that allows a cell to mate has a cost. And so during evolution, we'll see loss of the ability to respond to a mating partner. We can get cells to elicit a mating response without actually having a mating partner. So we can trick them into thinking there's an alpha cell by just adding alpha factor pheromone to the medium. In addition, we engineered into some of our strains a fluorescent reporter such that when they turn on this mating response, they'll turn green. And so then if we have a mixed culture that's mating competent and sterile, we can identify those sterile mutations by just adding alpha factor to the medium and counting how many cells turn green and how many remain dark. And so we have a phenotype sterility that we know is beneficial if it arises in long-term evolution experiment. And now we have a way of tracking visually in a high-throughput manner the fraction of sterile mutations within a population. In addition, there's sort of one more wrinkle with this idea of sterility, is that the cost can be modulated. And so if we take two yeast strains that are isogenic other than a single position in this signaling pathway, the mating pathway, we can have it such that they gain about 0.5% fitness advantage by becoming sterile, or they could gain a 1.5% fitness advantage for becoming sterile. And so we built these strains, and we used them in our experimental evolution paradigm. And so we initiated 300 populations that we call the R populations that get a 0.5% fitness advantage for becoming sterile, and 300 that get a 1.5% fitness advantage for becoming sterile. We propagated them under such a regime where we did a 1 to 1,000 dilution every 24 hours. And by doing that, we could then we went around this cycle 100 times to give us 1,000 generations total of experimental evolution for these 600 populations. During the course of this experiment, every 40 generations, we took an aliquot of these cells and we froze them down, so we have our frozen fossil record. And we also exposed an aliquot of cells to alpha factor and ran them through the flow cytometer. And this would tell us very quickly what fraction of sterile mutations were in each of these 600 populations. And so here's one of those 600 populations. So across all 600 populations, we were able to detect these sterile beneficial mutations in about a third of them. It was like 221. The simplest possible dynamic we could see is this. You wait around for some time, a beneficial mutation arises. This is the percent sterile on a log scale on the y-axis. It increases in frequency and then it fixes. So this is your classic selective sweep. This, however, was rare. Of the 221 times we saw these beneficial mutations arise, they only fixed 8 times. Most of the time, the dynamics look fairly complex. And so this is the most common outcome, is that the steriles will arise, they'll come up to some frequency, they'll make a U-turn, they'll go down and go extinct. And so this is clonal interference. The interpretation is that since this is an asexual population, you had one beneficial mutation that was sweeping, but another more beneficial mutation arose, outcompeted it, and forced that lineage out. We also saw more complicated dynamics, like steriles come up, they drop out, they come back up again. Or steriles arise, they come up, and then they get to some frequency, and they just stay there flat for hundreds of generations. So one of the points I wanted to make on this slide was that selective sweeps are rare. There's probably a lot of beneficial mutations, so there's lots of clonal interference. The other main point I wanted to make on this figure is that these sterile mutations move through the population faster than we would have expected. So I told you a couple slides ago that the benefit of these sterile mutations is either 0.5% or 1.5% by themselves. Yet, if you look at the slopes that we've measured, in all cases, these sterile mutations are moving through the population faster than we would have expected if they were just a sterile mutation. And so our interpretation is that when we see sterile mutations in these populations, it's not just a sterile mutation, but it's a sterile mutation piggybacking on some other beneficial mutations as well. As I said, we're going to try to keep ratcheting up the detail with which we can see the dynamics of adaptation. And so this is sort of the next sort of level of ratcheting up the level of detail. So this is a simple population where here we had a sterile mutation arise around generation 150. It peaked around generation 400, and then it went extinct. And based upon what I was saying on the last slide, we can make 2 predictions. First, because of the initial rate at which this sterile beneficial mutation spread, that it was not just a single beneficial mutation, but was multiple beneficial mutations piggybacking on one another. And the second inference is pretty obvious, is that because this sterile lineage failed to go extinct, there must have been some other more beneficial mutation that swept through the population. So to identify these other mutations, we sequenced this population at 12 time points, which is roughly every 80 generations. And when we sequence it, we can see all mutations that were present in that population above, say, 10%. And then we get a picture that looks like this. Again, we can see our sterile mutation in orange, rising around generation 150, peaking around 400, and then going extinct. We can also see, and so this on the top is the frequency of all mutations in the population. Down here is the same data, just plotted as a molar diagram. And so the width of these is indicative of the frequency of that mutation. So this gives you an idea of what mutations are falling in which background. So we can see that the sterile mutation fell within the background of a ROT2 mutation that was already sweeping through the population. So this was that mutation that the sterile mutation was piggybacking on top of. In addition, we can see a mutation in IRA1 also came out of the ROT2 background. The IRA1 mutation swept through the population and forced our sterile lineage to go extinct. So this was the competing beneficial mutation. So this is one example. We did the same whole genome, whole population time course sequencing on 40 populations. And so this is like the bird's eye view, or the view from 20,000 feet, or however high birds fly. And so, as I shut the door. I'm using mine, which shouldn't have that option. There we go. Well, we advanced. Okay. So this is sort of the view from space, or I don't know where we're viewing this from, somewhere up high. What I want to do in the next slide is sort of walk through a few of these populations just to give you some of the general take-home messages from this type of data. So we consider this are 40 sort of really nice case studies of the dynamics of adaptation. So one of the first things we see when we look at adaptation across these 40 populations is that selective sweeps are rarely single mutations or rarely even single phase events. And so this population sort of illustrates that nicely. Here you have a group of 5 mutations that we've called cohorts of mutations, mostly for lack of a better word, that move through the population more or less in lockstep, so that you can't really distinguish which ones came first. This is followed several hundred generations later by its cohort of 3 mutations. And sort of right on the heels of that, before this one is even fixed, you have this other cohort of 5 mutations. And so here you see mutations, more often than not, are not individuals, but are these cohorts of mutations moving through the population. Another general trend that we can see from this data is that we tend to see the same genes hit by mutation over and over again. So here you have a population where there's a single mutation in PDR5 that was moving through the population. It got outcompeted by this cohort of 6 mutations, which in turn got outcompeted by this cohort of 9 mutations. And if you look in both this orange and yellow cohort, there's a KRE6 mutation. So what this means is around generation 600, there were 2 different KRE6 mutant alleles. One was on its way up and one was on its way out. I mentioned to you earlier that we see some interesting dynamics where mutations will come up and they'll hang out at a certain frequency stably for hundreds of generations. So we sequenced a few of those populations. So here's a population where we have a bunch of mutations hanging out around 80%, a bunch hanging out around 20%, and then we can actually see selective sweeps happening within one of these 2 stable subpopulations. And so I think from looking at this type of data, we can get a good picture of the dynamics of adaptation, some sort of general principles of adaptation in these populations. But this doesn't tell us anything about which of these mutations is actually driving adaptation. So to get at that, we can come back to this observation here, that we tend to see the same genes hit by mutation over and over again. And so in total, we identified over 1,000 mutations. 723 of those mutations actually hit a gene. And so if you do sort of the randomization of just throw 723 darts across the yeast genome, which contains about 5,800 genes, you would expect that most of those darts would hit a unique gene, but that you would hit 20 genes or 40 genes twice by chance, and only twice would you hit a gene 3 or more times by chance. What we see, however, in our observed data is a very skewed distribution where we have 24 genes that account for a disproportionately large number of the observed mutations. So these 24 genes are likely the candidate genes in which drivers of adaptation are occurring. So just to drive that point home, these 24 genes represent about 0.6% of the yeast genome by size. They account for 14% of the mutations we observe. So what are these genes? So these are the 24 genes that we can say are statistically overrepresented in our mutation analysis. And so these are likely candidates for drivers of adaptation. Leading the list is IRA1, which we see mutated 21 times across these 40 populations. We can see a lot of, we can look at what biological processes tend to be hit. We see a lot of mating, which is not actually a surprise since we sort of set this whole experiment up to track sterile mutations as they move through the population. We also see, like IRA1, a lot of negative regulators of RAS, and we also see a lot of things involved in cell wall assembly and cell wall biogenesis. Of the 141 mutations that occur in these 23 putative drivers, only one of them is synonymous. There's a very high DNDS, and that's compared to 18% synonymous for all other mutations. And so when these genes are being mutated, likely their function is changing. So again, that's sort of additional evidence that these are probably drivers of adaptation. Because we've sequenced them all, we know exactly how they're changing as well. So these are the 21 different mutations that arose at IRA1, and you can see that it's missense, nonsense, frame shift. So this suggests that, at least for IRA1, we're likely selecting for loss of function of this protein. That's in contrast to the cell wall assembly genes like KRE6 and GAS1, where we only see missense mutations. which suggests that at these loci at least, we're selecting for alteration or attenuation, but not loss of function. And if we actually go in and make the deletions to GAS1, the cells are sick, and I think KRE6 is an essential gene, so we can't make that deletion. And so this type of sort of statistical analysis gives us a good idea of what are sort of the major genes that are drivers of adaptation. But we want to take this sort of one step further and ask not just what are the likely drivers of adaptation, but what are the fitness effects of all of these beneficial mutations that are arising in these populations. And to get at that, we have to go back to sort of what the yeast cerevisiae community says is the awesome power of yeast genetics. And so our strategy for quantifying the fitness effect of all the mutations that arose within an evolved lineage is to do the following experiment. So this is a cartoon of evolution. You accumulate mutations. This strain has accumulated 5 rainbow-colored mutations. We can take this strain, we can back-cross it to a MAT alpha version of the ancestor. This will produce a diploid that will be heterozygous for all of these evolved mutations. And we actually set this up such that this diploid is also heterozygous for the dominant drug sensitivity gene CAN1 at 2 unlinked loci. We then use some yeast genetics tricks. We convert this MAT-A alpha diploid to a MAT-AA diploid, so we get rid of all of the mating type information for MAT alpha. We then trick this diploid to sporulate by complementing the missing MAT alpha information on a plasmid, and we plate them to selective media that contains the drug cannabinine. So this will kill all of the diploids, and in fact it will also kill 3/4 of the haploids that have a functional CAN1 gene. But what this allows us to do is to put on we use these large auger plates to get essentially an arbitrarily large number of MAT-A progeny that are almost essentially isogenic to the evolved strain, other than that they have random combinations of evolved mutations. And so now we're essentially using Mendel to kind of shuffle the deck and generate all possible combinations. And the pools we generate tend to be on the order of 10 to the 5th to 10 to the 6th individuals. So we can get very large pools of random segregants from this. So once we've essentially broken up the linkage of all of our evolved mutations, we can then take this pool and we can put it into one tube, or in our case, one well of a 96 well plate. And then we can propagate this pool forward exactly as we did for the evolution experiment. And as we do that and we expose it to selection, we're going to get changes in frequency of these individual mutations. And we can look at the rate at which the frequency changes as we propagate this pool forward. And from that, we can quantify the fitness effect of all of the mutations that were in the background of these evolved clones. So I'm going to give you a simple example. So here's a population that has 3 mutations that fixed by generation 545. And so here in gray are the ones that were not in the clone we picked to backcross, but the ones in color are. So we took this clone from generation 545. We generated a large pool of segregants. We then propagated that pool forward and looked at the change in frequency of all of these alleles in the pool. You can see ELO1 basically stays flat. So this is a neutral mutation. It didn't enrich in the pool. But both sterol12 and GAS1 increased in frequency. And the rate at which they increase is their background average fitness effect. And so this is a really quick way of measuring the fitness of all of the mutations that were in one of our evolved strains. This would have been easy to do individually because there's only 3 mutations, but it was really a test case. We also did this with more complicated ones like this. In this case, there, I think, are 14 mutations, so there would actually be sort of a lot of reconstruction that would need to be done to measure these individually. But we just took this, we back-crossed it, we got a large pool of segregants that had random combinations of these evolved mutations, and we propagated that pool forward over time, and this is what these results look like. Immediately, you can see that most of these lines are flat, which means most of these mutations in this evolved clone are neutral. If I highlight only those that are significantly different from 0, you can see that there are three major driver mutations: ROT2, IRA1, and MID2, and these are the fitness effects conferred by those mutations. And if we sort of map those back onto the dynamics, you can see that each of these driver mutations corresponds to the three main selective sweeps that occurred within this population. So at least for this population, it's consistent with the idea of when we see a cohort of, say, 5 or 6 mutations moving through the population, that it's one driver mutation and a whole bunch of neutral hitchhikers. So we did this same quantification, sort of back-crossing pool construction and quantification for 11 different pools. From those 11 pools, we were able to then measure the fitness effect of 116 mutations from these evolved lineages. And that's shown here, broken down by which clone we had chosen. And from some populations, we did clones at 2 different time points that had different sets of mutations. And what you can see is we found, I think it was 24 mutations that had fitness effects different from 0. We found one and only one possible deleterious mutation. And we're actually not even sure that this was a deleterious mutation at the time it arose. It's a stop codon read-through of GCN2 involved in stress response. And we're actually interested to find out if maybe it was beneficial in the context that it arose. But here, overall, we show that it's actually deleterious. We also don't find any evidence for intergenic or synonymous mutations that have any effect on fitness. I want to come back to this idea that I mentioned earlier about how we can sort of statistically look for mutations that we see more often than expected by chance and say these are our list of putative driver mutations. Well, now we've actually validated 116, and we can ask how well these sort of statistical approaches do. And so before, our threshold was that if you were mutated 2 or more times, we called you a beneficial mutation. And so everything that's in a solid red circle, we had known to be a beneficial mutation, and we validated that here. But these open circles are true beneficial mutations that we missed by these statistical approaches because they were mutated only 1 or 2 times across these 40 populations. And you can see that these are often sort of low fitness beneficial mutations that were missed because they just didn't occur enough. But interestingly, 2 of the largest beneficial mutations were missed in our statistical approach. And so this is HSL7 and KEL1. I just want you to keep those 2 in mind. We'll come back to why we missed those in about 2 slides. Maybe 1 slide. I can't quite remember the progression. Another thing that we can do is we can, if you imagine there's no genetic interactions, that all of these mutations just add together and everything's perfectly additive. Then if we take all of the mutations, the fitness effect of all the mutations in the background of this BYS1E3, it should add up to give you the fitness of that clone. And so we measured the fitness of all these clones, and we just said, is that the same as the additive expectation, just putting all these mutations together? And so this is the additive expectation of just summing across all the mutations in each clone. And this is the actual measured fitness of that clone. This orange line is the unity, so where we'd expect everything to fall. And for the most part, we see a pretty good trend here. There's one clear sort of outlier of that trend, and that's this point here, where when we measure the fitness effect of the mutations individually, we can't recapitulate the fitness effect of the clone. And this population looks fairly unremarkable by itself. You had a single sterile mutation that was moving through the population that got outcompeted by this cohort of 11 mutations that sort of abruptly came up and swept the fixation. Interestingly, if we start looking at what mutations were in this cohort, we'll again see what I just showed you on the previous slide, HSL7 and KEL1. We'll also notice that there's an enrichment for non-synonymous mutations in genes that all localize to a similar place in the cell. And so this is actually the first time we've seen some biological function enrichment in these cohorts of mutations. And so we went in and we made the specific reconstruction of all of these mutations. And we find that there are 2 mutations that interact significantly. And those are HSL7 and KEL1. So by itself, KEL1 is neutral. And HSL7 is a modest effect beneficial mutation, maybe about 3%. But when you put them together, you get a really large, something like 8.5% fitness effect. And so this is a case that lots of evolutionary biologists are looking for of synergistic epistasis, where the combination of two mutations is greater than the sum of their individual parts. And so we were excited to find this. It was interesting. And then we started looking back, and you can notice that these mutations, we can actually see some of the mutations at earlier time points before they really took off. We noticed that, surprisingly, the KEL1 mutation arose several hundred generations before the HSL7, which means we had a neutral mutation, and that neutral mutation hung out at very low frequency until this HSL7 mutation arose within its background, and then it and everything else with it swept to fixation. And so one of the projects that we've very recently started in the lab is to ask if this KEL1 has essentially opened up a path to higher fitness that was not easily accessible from the starting strain. And so one of my grad students went in and restarted this population from generation 270 and generation 370, so before and after the KEL1 mutation arose. And he once again followed this sterile mutation, which, as you remember earlier, we can track phenotypically. And we wanted to ask if this population does anything like it did in the first iteration. And so I'll show the slide, then I'll kind of walk you through the results. So before the KEL1 variant arose, so starting from generation 270, these different green traces are following this sterile 12 lineage. In many of the cases, the sterile 12 lineage in the replay actually ends up fixing. But in many cases, the sterile lineage still continues to go extinct. Then here are the restarts at generation 375 after the KEL1 mutation. And again, you see many of the cases the sterile 12 lineage won in the replay. But in many cases, it once again loses. What my grad student Ryan did is he took all the cases where the sterile 12 lineage again lost, meaning that some other non-sterile lineage swept to fixation, and he asked if it was the same KEL1 containing lineage. And the way we can check is that another mutation in this lineage introduced a restriction site. And so we could just simply say, of these cases where the sterile 12 lineage went extinct, how many times was the same low-frequency lineage that caused it to go extinct? And before the KEL1 mutation, none of them were the same low-frequency lineage, but after it, three out of these 48 replicates were. And so what Ryan then did is he went back and redid another 400 replay experiments from the same time point, and that was what was necessary to collect 48 different lineages where the same low-frequency lineage once again rose to higher fitness. And the idea is that we want to ask, do you keep finding the same KEL1 mutation in that background, or is it the KEL1 opens up a large number of possible beneficial mutations that were previously difficult for this population to access? So this was the original population, and this was the first replay that we sequenced. Shown in color are ones that were present in the original and also present in the replay. Of course, KEL1, IQG1, and this intergenic. We don't see HSL7, but we actually see HSL1 in this replay experiment now. And so we've now sequenced all 48 of these, and we're starting to look for putative genes that may also interact synergistically with KEL1. So here we're using an evolutionary replay experiment to identify genetic interactors of this KEL1 mutation. So this is sort of brand new. We don't have too much data, but it's one of the things that we're very excited about. So I'll talk a little bit more. I'm trying to sort of gauge how much time I have. One of the things that came out of the original sequencing is that we noticed that many of the mutations we were saying that are beneficial kind of look like they're loss of function. And so this is just if we look at what the classes of beneficial mutations versus neutral mutations. That neutral or intergenic and synonymous, whereas beneficial tend to be like nonsense and frame shift. Like we're really destroying protein function. And so one of the simple questions we had is the fitness that we're seeing recessive? And so we did what I thought was a kind of a real simple but clever experiment. So this is 12 haploid evolved lines. We simply cross them to an ancestral wild type. And we even controlled for mating type by switching these to MAT AA. and we showed that when we mask all of the evolved mutations with a wild type copy, fitness basically declines, or in many cases even goes away. Interestingly, this population here, where fitness still stays pretty high, is the population that had this KEL1 HSL7 mutation. So we think that that's actually probably a gain of function or alteration of function mutation. And so this type of result, that many of our beneficial mutations in haploids are recessive, kind of begs the question, like, why didn't we, what would happen if we would do the evolution experiment in diploids? And so we did that experiment. We evolved 48 diploid populations for 4,000 generations. And then after 2,000 generations of evolution, we sequenced 24 clones to get an idea of what types of mutations are occurring in the diploids. And we had really two predictions going into this. One is that if most beneficial mutations that haploids are getting are recessive, that diploids ought to evolve more slowly, and that when diploids get beneficial mutations, the spectrum of those mutations should look different from haploids. So the first of those results is shown here, that this is the haploid populations I showed you before. The diploid populations do in fact evolve more slowly. When we sequence evolved diploids, we also find that they get different types of point mutations. And so this is the list I had showed you earlier for the types of mutations we see in haploids. This is a similar table for diploid mutations. And you can see that there's some different biological processes. For instance, we don't see mutations in the mating pathway, which is not a surprise because in diploids this pathway is normally shut off. We also don't see negative regulators of RAS in diploids. It's not quite clear why. Maybe it's just hard to access these mutations, or maybe there's something different about the RAS pathway in diploids. We do see cell wall assembly and cell wall biogenesis in both, but even within this biological class, we see some genes like carry6 in both haploids and diploids, but we see carry5 only in the haploids, and we see CTS1 only in the diploids. And so even within a shared biological target of selection, there are different genetic targets of selection in haploids and diploids. This other column here, this sort of half moon or full moon, is whether or not the mutation that we've observed in diploids is heterozygous or homozygous. Most mutations are heterozygous, but interestingly, a lot of the driver mutations, such as ACE2 or CTS1, often come up as homozygous. And to get some pictures to what's going on in these populations, we went back and did the same whole genome, whole population time course sequencing now on some of our diploids. And so this is an example of a diploid population. You see a lot of mutations that come up and hang out at 50%. These are the heterozygous mutations. And you can see a few mutations that manage to make it to being homozygous. So I want to strip everything away other than two mutations. This LTR that was initially heterozygous, sort of inadvertently when we built the strain, there was a few heterozygosities, including LTR1, as well as ACE2. You see this ACE2 mutation arise. It increases, and right around 50%, there's kind of this little shoulder before it continues on to sweep. And when ACE2 sweeps, it brings with it this LTR as well. And so this suggests that this was probably a loss of heterozygosity event that not only made ACE2 homozygous, which is on chromosome 12, but also the nearby LTR. And so to test that, we went in and we genotyped a bunch of individuals from this population before the sweep, halfway through the sweep, and at the end of the sweep. And what we find is that before the sweep, all of the individuals are homozygous wild type for ACE2. At the end, they're all homozygous mutant. That shouldn't be surprising. But at this 50% inflection point, the whole population is basically heterozygous. And so this is consistent with the model of an ACE2 mutation arising, the heterozygote sweeping through the population. You then have loss of heterozygosity, which then itself sweeps through the population. And that loss of heterozygosity event encompass not only ACE2, but also the LTR on the same chromosome. We've also begun looking at what might be some of the explanations for these diploid-specific and haploid-specific mutations that we see. So this is the last point I want to make. And so we reconstructed some haploid-specific and diploid-specific mutations. So CTS1 was diploid-specific. When we put it in a haploid background, there's no fitness effect, which explains why we don't see it in haploids. But we find that it's actually beneficial, and it's almost equally beneficial as either heterozygote or homozygote in the diploids. So it's actually a bit surprising because we actually only see it as a homozygote in the diploid, and it's still something that's a little surprising to us. If we look at some haploid-specific mutations, I'll go through KRE5 first. So it's beneficial as a haploid. If we can make it as a homozygote diploid, it would be beneficial, so that's shown in blue. But it never arises and never gets to be a homozygous diploid because it's neutral as a heterozygote. We can't find a significant fitness advantage. GAS1 is kind of similar. It's beneficial in a haploid, and it could be maybe slightly beneficial if we can make it a homozygous diploid. But the heterozygote is actually quite deleterious. And so this suggests that there might even be an incompatibility between this evolved mutant allele and the its wild type version. And then sort of the obvious one is that for something that we see in both haploids and diploids, no matter how we reconstruct it, it's either haploid, heterozygote diploid, or homozygote diploid, it's beneficial. And so most of what I've told you about has been the simplest possible experiment we could do is just either haploid or diploid yeast in rich glucose media at 30 degrees asexually. What my lab is starting to do now is to sort of turn all these knobs that we have at our disposal. And so we're doing evolution in different ploides, in different species, in different strains. We're tweaking environments so that we can do evolution in sexual populations. We're doing shifting and alternating in environments. We could do the experiment where you sort of ramp the incubator up 1 degree every 100 generations and ask, can evolution keep up with that change? We can change how we, or more appropriately, how the robot does the dilutions to introduce migration between populations or to vary fundamental parameters like population size. And of course, we have at our disposal all of these really nice tools, the frozen fossil record, being able to quantitatively measure fitness and to immediately see exactly how all of our populations are changing at the genetic level. So just to summarize, we're sort of motivated by this question of reproducibility in evolution, that if you run the exact same evolution experiment from the same starting point, how reproducible, sort of what's the distribution of evolutionary outcomes. I mentioned to you that the first observation we had is that sterility confers a fitness advantage. And then we can use this as a tool to watch this phenotype emerge in the context of experimental evolution. I then told you about how we sequenced a large number of these populations using whole genome, whole population time course sequencing to get a picture of the dynamics of adaptation. From these case studies of adaptation, we've begun to look at the distribution of fitness effects of individual mutations using what's really sort of a QTL, quantitative trait mapping-like approach. From this, we've revealed things such as epistasis amongst some genes within some of our populations. And then at the end, I kind of went quickly through it, but we are now starting to look at these different situations, such as can we directly compare adaptation in haploids versus diploids. And so with that, I'd like to thank my lab. So I have 1 postdoc, 3 grad students, and a few undergrads, various funding sources, people with technical help with flow cytometry and sequencing, and thank you for listening. Mm-hmm. Yeah, that's a really good question. Is that because you sort of get this diminishing returns as you get closer to the optimum, haploids have a fitness advantage. If you just construct isogenic haploids and diploids, the diploids have somewhere a 3 to 5% fitness advantage over those haploids. And so the question is the rate of adaptation, the difference in the rate of adaptation due to recessive beneficial mutations or due to just distance to the fitness optimum? So one of the things I can say to that is that the fitness that haploids gain is greater than the distance between them because they adapt something like 8% over the first 1,000 generations, but the difference between haploids and diploids is 3, maybe 5% at the most. And when we actually do the reconstructions, we find that a lot of the beneficial mutations in haploids are in fact recessive. And sort of at the end, I was kind of going through and showing how some of these would be inaccessible to those diploid populations, even though if you could make them as a homozygote, they would provide a fitness advantage. So I think certainly proximity to the fitness optimum could play a role. But we think a lot of it is driven by just not having access to beneficial mutations, which in this case is often just throwing overboard everything you don't need. Yes. Okay.
When you were distinguishing drivers as a piggybacker,
Uh-huh.
did that have the analysis have the ability to identify sort of paired interactions like you saw with the KEL1 and the HSL?
Yeah, so when we were doing the back crossing and we would get all these segregants, we'd measure the fitness effect of them all individually. One of the first tests we would do is add up those, because those are really background average fitness effects, because we've made all possible combinations, all possible genotypes, and so we've measured the fitness of, say, the HSL7 averaged over all backgrounds which can occur. And if there's no epistasis, so the no model of no genetic interactions, everything should add together and it should give us the fitness effect of the evolved clone. So that was our first test. So how sensitive we are is not clear because we expect there to be some epistasis. Lots of labs have found there's just generic diminishing returns epistasis, which is like this progressive tax that beneficial mutations have to pay when they fall in favorable backgrounds. And so we think we are at least able to pick out strongly interacting mutations. Whether or not there's any weakly genetically interacting mutations, we're not sure. We have looked at when we see these groups of these cohorts of mutations, are there biological processes that are overrepresented in any of them? And by and large, they look like they're just random subsets of genes plucked from across the genome. So yes, we think we have some power. It's not that great of power to detect interactions this way.
Is there any natural population who also have undergo similar episomal change? If so, if any, do they undergo the same genetic?
So the genetic changes we tend to see in laboratory experimental evolution look, there are some ways they look like natural variation. So IRA1, which is a major hit we find, is also a major hit from QTL studies. So in some ways we hit similar things to natural variation. But by and large, I think what we see in laboratory experimental evolution is very different than the kind of mutations we see out in nature. The question I often get is, why aren't yeast sterile in nature? And it's because there's pleiotropy, that if sterility provides a fitness advantage if you're growing asexually, but if you're ever asked to mate, that's a lethal mutation. And so I think a lot of what we see in laboratory experimental evolution is more akin to like what you would see in like cancer evolution or something. And they're like, we see some rearrangements and we see things that would be deleterious in the natural environment. And so I think it's a great model system for studying dynamics and basic rules of evolution. It's not the best model system for saying how evolution should be happening to yeast in nature. Yes.
I might have missed this, but I think you said about 6,000 mutations that you had in the sequence, and you had about 700 in yesterday.
Yeah, it was 1,000 in overall and about 700 in the coding sequence.
Okay, so in the, did you have any that were enriched in particular in the beneficials, and did those correspond with the genes that you thought as associated?
Yeah, so we've looked a little bit for intergenic regions that are overrepresented in a similar way to the gene. And we had not found anything. And we are right now in the process of, so we've now done this with diploids, and we've done a whole bunch of lines that were haploid that became diploid as well, which was kind of surprising. And we think from that data set, we have a lot more mutations. So I don't know what we have, probably close to 7,000 mutations from those two data sets. And we think in diploids, we may have a better chance at finding regulatory mutations. And so there we're embarking on a similar study where we basically ignore all the coding sequence and we look at intergenic regions and ask, are there any intergenic regions that are overrepresented? And there's a few that may be potentially interesting because we see homozygous mutations in multiple lines in similar intergenic regions, and that is kind of unusual since most mutations in diploids are heterozygous. So we're just embarking on really doing kind of a meta-analysis of all of our data plus whatever data is out there from other labs to look for intergenic regions. Because what we would love to find are intergenic regulatory mutations that have a fitness effect, synonymous mutations if they have a fitness effect. Despite looking for them, we haven't found any yet. Yeah, absolutely. And so the mutations we see in the RAS pathway, when people ask me and I kind of wave my hands, that it's really about shifting conditions. And so as long as we faithfully dilute them back into fresh YPD every 24 hours, those RAS mutants are fine. What they're doing is they're essentially taking their foot off the brake when they're entering stationary phase, and they're just eking out a few more divisions. Now, if we wouldn't dilute them back and they just hang out in stationary phase or encounter some other stress because we've shifted the environment, those would likely be deleterious or even lethal. And so, Gavin Sherlock at Stanford has done some experiments to try to show that a lot of the mutations we see they affect different parts of the growth cycle, and if you extend the growth cycle, they actually are deleterious. And so, absolutely, if we were to shift conditions, there would be many cases where the mutations we see would not be favored. Luis.
You showed one of these molars plots that had two coexisting populations.
Mm-hmm.
How often do you see that? And what's going on?
Yeah, so the coexisting populations, we were really excited about. We're like, oh, this is really cool. They must be like cross-feeding each other, and it's like, it turns out like it is frequency-dependent selection. There is balancing, but it's kind of boring. Essentially, when we transfer them, we don't shake the plates, and so you get these mutations in this ergosterol pathway that cause yeast to stick to the plastic sides, and they also fall in the middle, but when they fall on the bottom with the ancestral strain, they lose to the ancestral strain. But where they're stuck up on the wall, there's no ancestral strain, so they essentially can do the spatial structuring. And then we mix the whole population before we transfer them, and the ancestral strains fall to the bottom, and then the erg ones stick to the side. And the simple test is if you do the same competition, but you shake the plate, spatial structuring breaks down, the erg mutations go extinct. And so it wasn't a cross-feeding. It wasn't particularly exciting. It is common. It's actually kind of annoyingly common because it makes it difficult to do fitness assays on populations that have frequency-dependent mutations. So we see it. We acknowledged it, but we don't really care to see it that much. It's kind of interesting that you can get some semblance of ecology in a very simple well of a 96-well plate and some spatial structure.
Okay, that's great, yes.
Thank you.